Today we are going to discuss about cell membrane. The cell membrane is the structure which separates the extracellular fluid and the intercellular fluid. This is the cell membrane. In an organ, the extracellular fluid and intercellular fluid are separated by cell membrane. The main component of cell membrane is phospholipids, and the structure of phospholipid is one hydrophilic head and hydrophobic tail. The hydrophilic head are polar and the hydrophobic tails are non-polar, and the structural like this. The hydrophobic heads are towards the external extracellular fluid and the one hydrophobic heads are towards intercellular fluid. The external which are faced towards the extracellular fluid are called outer leaflet, and the structures which are facing towards the intercellular fluid, these are inner leaflet. This is phospholipid bilayer. One is outer leaflet, one is inner leaflet. First, we will consider about outer leaflet. The outer leaflet is one hydrophilic polar head and hydrophobic non-polar tail. This one, outer leaflet, hydrophobic polar head and hydrophobic non-polar tail. And in inner leaflet also, that is hydrophilic polar head and hydrophobic non-polar tail. Here in inner leaflet, this is hydrophilic head and hydrophobic tail. These compounds are not permeable to highly charged compound because these are polar, so highly charged compound are repelled from the extracellular fluid. Also from intercellular fluid, highly charged compounds are repelled. The cross it show the highly charged compounds are cannot pass through cell membrane, and high molecular mass also cannot pass through cell membrane. Like insulin cannot pass through cell membrane, but thyroxine thyroxine T3, T4 are very light-weighted compound. It can pass through the cell membrane to the intracellular fluid from extracellular fluid, but insulin cannot. So small molecular mass can pass. Lipid soluble compound can also pass because the inner surface, these layers are lipid soluble. So if a lipid soluble compound can mix with the cell membrane and then pass towards the intracellular fluid. But phospholipids are outer leaflet. The phospholipids of the outer leaflet, these structures, these boxes are made up of mainly two types of phospholipid, the phosphatidylcholine and sphingomyelin. So in the inner leaflet also has two different phospholipids. These are phosphatidylethylanolamine and phosphatidylserine. So the outer structures, these structures are different, and these structures are also different by their structures. Outer leaflets has many carbohydrate molecules, but these outer leaflets have many carbohydrate molecules, but inner leaflet has not any carbohydrate molecule. So they are very different from each other. So these are asymmetrical. And in another point we will learn between the this is the cell membrane hole, and this is the outer leaflet, and this is the inner leaflet. In this inner leaflet, these are the tails and these are the heads, but these compounds which are intercalated between the structures of tails is called cholesterol. These are intercalated inside the cell membrane. The phospholipids are not fixed. It can slip laterally. It is called fluidity. It means here the structures are not fixed. It can move side by side. So this movement is called fluidity. Fluidity helps in mainly three processes: exocytosis, endocytosis, and biogenesis. Exocytosis is if the cell is there, one compound is inside it, and we have to throw it outside. In endocytosis, there is one component here, we have to take it inside. Mainly this is for WBC in case of phagocytosis. If one microbe is outside and we have to uh, confirm one pseudopodia like structure, then we have to engulf it. This is called endocytosis. In biogenesis, one cell is, let it is the cell, then it is divided to two small cells by mitosis, then it will then grow through mature cell. So this growth is called biogenesis of cell membrane. These structures are growing, so we will discuss it clearly. Let's take exocytosis. In first case, there is pancreas. The beta cell of pancreas secretes insulin. Let's take, this is the cell of pancreas, and here the insulin secreted from the inside the cell membrane. We have to take it to the blood vessels. So we have to take, it will come to the cell membrane, then it dissolve with the cell membrane, then it go outside the cell membrane, then this fracture side of the cell membrane will repair itself due to the process of fluidity. So the fluidity, due to the fluidity, many phospholipids come due to side by side, and it can repair the cell membrane and it will become secure. In case of endocytosis, let's take one WBC in phagocytotic cell, and one pathogen is outside the cell. Then it will create one pseudopodia, then it will engulf it. So to create the pseudopodia and engulf it, to take it inside, the cell membrane needs more flexible structures. So these flexible structures can possible only due to the fluidity. Next, biogenesis. Let's take, here is the cell and we have the cell, then it becomes mitosis, then it will growth its size, then it will next go for another mitosis. So here in the growth of a cell, the cell membrane size also increases due to growth. So due to the increase of sizes, the cell membrane shall prepared inside the cell, so it will go to the cell membrane. Let's take, here is the small cell membrane and here the cell membrane are prepared, then it will go to here, then these structures are intercalated between it because it has fluidity. So from here it are cleaved and go separate side by side, and this will enter into this side, and it will cause no lack or gap in the cell membrane structure. The fluidity mainly depends upon temperature. Temperature increases, the fluidity also increases. Commonly, the increase in temperature, we know if a gas is here or any molecule is here, if temperature increases, then their movement also increases. Like this, remember things that fluidity also increases if temperature increases. If cholesterol increases, then fluidity decreases. Simple reason. If here cholesterol amount is 1, then it can move freely. But if here is a cell membrane and here 1 cholesterol, 2 cholesterol, 3 cholesterol, 4 cholesterol, 5 cholesterol, 6 cholesterol, many cholesterol there, so it is very tightly packed, so it cannot move, so fluidity decreases. Temperature increases, fluidity increases, and cholesterol increases, fluidity decreases. Structure of fatty acid. If here the structure of fatty acid, it means here is the head and here is the tail. This tail structure, if these are straight, then these are less fluid because they are saturated. If these structures are bent, it means these are unsaturated and these are more fluid. If they are saturated, they are straight and they are less fluid, and if they are bent, they are more fluid. Just remember, if one has straight leg, then he can move straightly. But if one has bent legs, they can move here, there, like this. So if they are straight, they are saturated, they are less fluid. If they are unsaturated, they are bent, they are more fluid. Then we'll learn about lipid raft. Lipid raft is the same cell membrane structure, but it contains one receptor. Here, this red color one was receptor. This is serpentine receptor or 7-pass receptor. Here, there are some characters. Phospholipid are more tightly packed. Here, see the phospholipid 1, 2, 3, 4, 5, or here 5. These are very tightly packed phospholipids, and fatty acid components are longer. These fatty acid components are, these tails are longer than other fatty acids. And tightly packed cholesterol. I, 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 I taught you about here the tightly packed fluidity decreases. Here also tightly packed, so fluidity can decrease. Tightly packed cholesterol. Here the cholesterols are tightly packed, and fatty acids are more saturated. Means here saturated means straight. So these fatty acids are more saturated means more straight. Understood? And then the receptor proteins are present. Here the receptor proteins are present. Here the compound like insulin will come, then it will create some changes in the cell. Receptor is a protein structure which here is the cell membrane, here is extracellular fluid, here is intracellular fluid. If insulin comes or some other hormones comes, here it will bind here. Then if a protein structure bind to one end with a hormone or compounds, and it can change some, it can lead to change some structures or RNA synthesis or protein synthesis inside the cell. It is the receptor. The proteins associated with the cell membrane, these are cell membrane. Some proteins are also associated with it. Proteins are made up of 50% of the cell membrane. 50% of the cell membranes are protein, and 47% are phospholipids, and 3% are carbohydrate. Then the protein associated with cell membrane. Here we learn about 50% are proteins, 47 are phospholipids, and 3% are carbohydrate. The proteins are around mainly divided into two parts, integral protein and peripheral protein. Here, if these blocks are cell membrane, then this is integral protein. These are occupied the space from one side of the cell membrane to outside. These are outer leaflet, these are inner leaflet, these are extracellular fluid this side, and this side intracellular fluid. So these integral proteins occupy space from one side to another side, but the peripheral proteins are just either the outside of the cell membrane or inside of the cell membrane. These are very loosely attached with electrostatic force. It can just detach from the cell membrane by change of some pH or temperature. Just small forces it can detach from the cell membrane, but integral protein cannot. Let's teach about something, how much type of, let's learn about how much type of these proteins are. The integral proteins are central water channel, receptor, enzyme, carrier or linker, and the peripheral proteins are cytoskeleton, or sometimes it may be the receptor. Let's learn about first integral proteins. Integral proteins, will first learn about central water channel. The central water channel is one protein structure. This one is protein structure. These are the cell membrane layers. Here the inner gap was filled with water, and these structures are peptido polypeptide chains, which are composed of amino acids. Lots of amino acid will compound polypeptide chain, and this will comes into structure and create a water filled channel. This can, any compound can move from here to here to the cell. Then we learn about receptor. Let's here, it is the cell membrane. this orange one, and this black one is receptor. Let's here some hormones will bind. Let's take insulin, and then the action inside the cell will occur. This is also integral protein. See here, one side and two side, both sides are integrated with the cell membrane. Here enzymes, some enzymes, not all enzymes, some enzymes are integrated with the cell membrane. It will convert one things to another thing. Like the definition of enzyme, it will convert some substrate to product but not change itself. It is also sometimes integral protein, sometimes carrier protein. It's important to understand. Sometimes here see it's the structure of carrier protein. Let's take it's the structure of carrier protein. One thing's bind here, and then it changes the hand or something. You must understand the hand or any part of the structure to inside the cell membrane. Here it will take from extracellular fluid to its structure to intracellular fluid. This process is called transformation. Here this is called transformation of protein. Here structure changes to take some hormone or some important things to inside the cell. And linker linker is also bind with the cell membrane from both sides. Like inside it is connected with actin for example, and outside connected with collagen. Here collagens will connected with actin. Actins are with no, these are cytoskeletons. This cytoskeleton will linked with collagen and it will increase the structural stability of the cell. It will increase the structural stability of the cell. Then we will go to peripheral proteins. Peripheral proteins are cytoskeletons. Like here we saw these are cytoskeletons which are inside the cell. These are peripheral proteins. This leads to structural stability. Here in linker protein, these inside cytoskeletons are connected with outer side of extracellular fluid components to increase the stability. Then receptor. Sometimes we saw in this figure receptors are here and it has some intracellular component or intracellular binding site or effector site. This effector site are sometimes connected with another proteins. This protein leads to some second messenger system. Let's take here one component A will attach, then it will go here A, it will convert, then this structure will effectively B, then B go to some drastical changes in cells. So B is second messenger. So these proteins can cause second messenger system. So these are peripheral protein receptor. So let's review our this chapter from the first. Here this is the cell membrane. Cell membrane structure which separates the organ or the cell from extracellular fluid to intracellular fluid. Main components are phospholipids. They are hydrophilic head which is polar, then hydrophobic tail which is non-polar, then phospholipid bilayer, outer leaflet and inner leaflet, both have hydrophilic polar head and hydrophobic non-polar tail, and these are highly charged compounds, cannot pass through. Cross means cannot pass through the structure, means cell membrane, and highly molecular mass cannot pass through the cell membrane. Small molecular mass can pass through the cell membrane. Lipid soluble compound can pass through the cell membrane. Phospholipid of outer leaflet. These are phosphatidylcholine and sphingomyelin, and inner leaflet phosphatidylethanolamine and phosphatidylserine. And outer leaflet has many carbohydrates. Outer and inner leaflet are asymmetrical. This is important. Outer and inner leaflet are asymmetrical. They are not similar. Sometimes there are some cholesterols inside the cell membrane, intercalated within the cell membrane. Then phospholipids are not fixed. They slip laterally. This character is called fluidity. Then fluidity helps in exocytosis, endocytosis, and biogenesis. Exocytosis, like beta cell secretes insulin, it goes out, it is called exocytosis. And endocytosis, like phagocytosis cell, captures some pathogen by creating pseudopodia. Then biogenesis. Biogenesis, like increase in cell size, the cell membrane size also increases. So this needs biogenesis or synthesis of cell membrane. Then temperature increases, this will increases the fluidity. Then cholesterol increases, decreases the fluidity. Then structure of fatty acid. Saturated, it is straight or less fluid. This is the head, this is the tail, this tail is fatty acid. If this is saturated, this is straight and less fluid. If it is unsaturated, this is bent and more fluid. Then lipid raft. Mainly this consists of receptor and straight saturated structures and more cholesterol. Then protein associated with cell membrane. Integral protein, central water channel, receptor, enzyme, carrier and linker. Then cytoskeleton and sometimes receptor. Integral protein, central water channel, polypeptide chains are made up of polypeptide chains, and this channel is filled with water. Then receptor, the receptors which are binds to both sides of the cell membrane. Then enzymes, some enzymes are also are integral protein. Then carrier, this carrier made some transformational change too. This is called transformation. This transformation can cause the outer hand to come into inner hand. So then linker is like collagen bind to the intracellular cytoskeleton or actin. These are one example. For structural stability, these are linker. Then peripheral protein, cytoskeleton. Here we understood how the actin works to increase stability. So this is structural stability. Then receptor. Some receptors are joined to the integral receptor inside the cell. So this activate the second messenger system. This this is peripheral protein. So here ends with the structure of cell membrane, and in next part we will discuss about the transport mechanisms in the cell membrane. In next part we will discuss how one component is transferred to another side, and from another side it will transfer to another side of the cell membrane. Goodbye, thank you.